Okay, so hello everyone. My name is Sara, and I'm here to tell you a bit about the work that I've been developing in my PhD. And I'll tell you a bit about the mechanical stability of noted ubiquitin C-terminal hydrolase L1, and I'll give you some insights that were obtained from molecular dynamics simulations. So let's start with a bit of context. I don't know if most of you know what knotted proteins are, but they are actually a relatively recent concept. For a long time, it was actually imposed that a structure couldn't fold in a way as to hold a knot in its secondary structure. And it was actually the development of a knot detection method in the year 2000 that allowed a screening of the protein data bank. And that ended up making like a boom of curiosity between the fold protein community. And as of today, we actually know that about 1% of the proteins that are in the protein data bank have some kind of knotted structure. And actually, having this number in mind, we can tell that they aren't very common. However, by some reason, nature keeps maintaining these proteins. And we assume that there must be some kind of role that they have to maintain this structure. However, a consensus hasn't been reached on the role of the biological role of knots. And this actually might be due to two facts. The first one is that most studies that are actually, there aren't a lot of studies regarding the subject, and the ones that do exist are mostly focused on a specific type of knot, and that's the trefoil knot, that it's the first one that you can see here. That, as you can tell, is quite simple when compared to the other ones that are on the other side. And so, when we realized all of this, we decided to take things a little further. And so we decided to study ubiquitin carboxy terminal hydrolase L1, and you will soon understand why I say that we are going to take things a little further. Okay, so ubiquitin C-terminal hydrolase, as the name actually says, it is part of the ubiquitinating enzymes family. And these are enzymes that are actually able to hydrolyze the ubiquitin adduct, and they end up countering the ubiquitination process. And as all of you know, ubiquitination pretty much involves the covalent additions to the lysine residues of target proteins. And therefore, this ends up working as a modification that is involved in both membrane trafficking and also protein degradation pathways. So now, why UCHL1 specifically? As I told you, okay, very important part of the ubiquitin-dependent pathway. But on top of that, this protein is actually one of the most abundant proteins in the brain. Overall, it actually ends up making 1 to 5% of the whole neuronal protein. And as if this wasn't enough, UCAL1 is actually also involved in both Alzheimer and Parkinson's. And this is actually due to the fact that UCAL1 has been found in Lewy bodies. And as once again, if this wasn't enough, UCAGL1 is also highly expressed in several forms of cancer. And it's kind of a funny detail that UCAGL1 has actually been found to be expressed in cancerous cells that in its normal behavior don't even express UCAGL1. And so we, after knowing all of this, we thought, okay, this is a really cool protein, let's study it. And now let's focus a bit on its structure. So UCAGL1 is quite a small protein. It only has 223 residues. However, even being small, it has one of the most complicated 3D knotted structure that exists, and that is the 5-2 or the Gordian knot. In this case, it actually is a shallow knot, and this is due to the fact that the knotted core is actually pretty big compared to both the N and the C termini that are looped under that make the knot. And on top of this, UCL1, just like the other members of its family, actually contains an unstructured loop that ends up restricting the access to its active site. You can see it's highlighted here, the active site, and this loop ends up constricting its connection. And this ends up telling us that most folded proteins actually can't connect to UCHL1 because they can't really reach this part that is under the loop. However, obviously, ubiquitin can connect to it because its C terminal actually doesn't have a structure, it isn't folded, and so it can connect to this part of our protein. So now, still in terms of structure, our protein is a cysteine protease, and it has a catalytic triad, and this one is composed of a cysteine, an histidine, and an aspartate. And as all of you know, in order for a cysteine protease to be in its active form, the cysteine and the histidine need to be closer together, around 4.5 angstroms. And as you can see by the zoomed part that we have here on the right, In the apostate, this doesn't really happen because the cysteine and the histidine are around 8 angstroms apart of one another. So we can tell right away that in the crystallographic structure that we have, our protein, our cysteine protease with its catalytic right is actually misaligned for catalysis. However, when we go from the apo structure to the complex one, when it decides to show, okay, you can tell that our histidine actually shifts a bit and it's way closer to the cysteine. And actually, in this case, we have an active catalytic triad. And having this in mind, we can clearly tell that it's the connection or the bound to ubiquitin that actually promotes the activation of our protein. And so, after knowing all of this, we decided to pretty much lay down two major goals. We wanted to use fully computational methodologies. What we wanted to study was the knot involvement in the mechanical stability and also in the function of our protein. And we also wanted to try to understand the role of the knotting in both the catalytic triad activation and also in the ubiquitin binding process. And so, in order to do so, we started by studying our system's stability in overall, and we did this by performing long MD simulations, and we started with two different systems, the APA one and the complex one. Here, I'm just showing some characteristics, like we have the root mean square deviation and also the secondary structure, just to clearly show you that our protein is quite stable throughout the whole simulation. And this actually makes us feel more confident going forward with other type of analysis. Because most of the time, of course, with MD, we do this type of analysis. In our case, we can't forget that we have something that we also have to take in account for, and that is our knot presence and therefore the knot stability in our simulation. And so, in order to study this, we use the software called Chemonot. And this allowed us to see how many residues were actually involved in the knot throughout the whole simulation time. And also, it allows us to see what type of knot is actually in our protein. In this case, identify that throughout the whole simulation times, we had a 5-2 knot, the Gordian knot. And here we can see on top in the apo structure that in the N terminus, we have around 5 residues that are involved in a knot, while in the C terminus, we have around 6 residues. When we move to the complex form, once again, in the C terminus, we have exactly the same thing, around 6 residues. However, when we look to the N terminus, the story is a bit different because the replicates, 4 of the 5 replicates actually start with the 5 residues, just like the apo structure. However, with the passage of time during our simulation, this ends up shifting from the 5 initial residues to 4. And probably if we extended these simulations, all of them would end up in the 4 residues. So we can clearly see, looking at this, that there is some kind of shift in the complex that doesn't happen in the apo structure. With these values in mind, we actually were able to plot the distribution of our percentages of knots during our simulations and with the number of threaded residues. And we can see that, of course, if we, just like I told you before, if we cut around 5 residues, that going forward, we abolish completely our knot. And moving forward, we actually decided to only focus on the N terminus of a protein because, as you remember on the first slides that I showed you, the N terminus is actually quite close to the active site. And so we decided to focus only on this part of our protein. And the next step was actually to produce truncated variants of our protein. So we decided to look at our output structure, and we started to pretty much cut or delete a residue one at a time. So we cut the first one, the second, and so on and so forth. And then we ended up with 7 new truncated variants. However, when we looked at a complex form, we decided to change a bit this approach, and we only did 2 truncated variants because we were trying to save a bit of computational cost. Then we chose the 4th and the 7th ones because they are, as you can see here on the left, these are the residues that are immediately before and after the knotted abolishment, if you can say so. After we produced the truncated variants, we simulated them just like we did before with the other structures. And then we were able to once again do this plot now with all the truncated variants. And what is really important to see here is that even when we start cutting our protein, the knot structure really maintains, meaning that, okay, I cut it 4 residues, maybe we were expecting that the 4th one would be slipping a bit or something like that. And in this case, we can see that that really doesn't happen. So the plot and the tendency is almost the same, even in the truncated variants. The next step we decided to take was to evaluate the number of active conformations that we had on our protein. Here we are looking once again to the apple structure. As I told you, the cysteine and the histine were quite further apart. But however, when we were looking at our structure, we were able to see that there was another histidine that was quite close to our cysteine. And you can see here that one is histidine 143. And so we decided to do a scatter plot with both of these distances, meaning the distance between this cysteine and the catalytic histidine and the other histidine. And what you can see here on the right, first we decided to define a cutoff of 5 angstroms, and meaning that what was here on the left of the 5 angstrom was considered as being active, and what was on the right was considered inactive. Here in the apple structure, you can see that we have around 15% of active structures, and actually all of them are from the same replicate. As you can see, they are all the same color. And once again, we can confirm that in the apple form, our protein is actually inactive. When we do the same, but for the complex form, we can see that this is quite different. And so here we have a way higher number of active structures. It's actually around 60%, and all of the replicates end up moving from one side to the other. Once again, we can say with some level of certainty that it's actually the bound to ubiquitin that induces this deactivation of our protein. We then ended up doing this not only for the wild type versions, but for all the truncated versions that I told you that we generated before. And this allowed us to plot this graph here on the right. Now, first focusing on the green line, first you can clearly tell that our error bars are quite big. And they are not also quite big. There isn't really a tendency in our results, meaning that we start with around 15% of active conformations, then it goes up, then it goes down again, it goes almost to 0, then it goes up again. So we can't really define a tendency here. And we wanted to see if we could define the tendency, because here in gray you have highlighted the area where the knot is still present, and in white is the part where the knot has been abolished. And we thought, okay, maybe we could define some relationship between the presence of knot and the number of active conformations. In the apple structure, we couldn't do it at all, as you can tell. However, if you look at the complex one, we can kind of see a tendency. We start with a higher number of active conformations, and then this number clearly goes down, and then kind of maintains its value throughout the presence of the knot or not. In this case, probably we will need more replicates to actually be able to tell if this tendency is real or not, just an artifact. And we will probably need to also end up building the other truncated versions also for the complex version of our protein. The next step was to calculate the binding free energy of our protein. Obviously, in this case, we can't use the apple structure. Doesn't make sense. So we use the complex wild type one, the delta 4 and the delta 7. On the first two, you can see, okay, the values are quite around the same tendency, nothing really strange. But however, when we look to the delta 7, we can tell that one of the replicates really has a weird behavior compared to the other ones. And when we looked at this, we tried to understand where this contribution was coming from. And so what we decided to do next was to calculate the interface area between our protein and its substrate, the ubiquitin. And if you look at the last graph, you can clearly see that this interface area ends up matching almost perfectly with the difference in the binding energy. And when we stop to think a bit about this, it actually makes a lot of sense, because in this case, we had already deleted 7 residues. So our protein really has a lot of space to move and to pretty much accommodate to ubiquitin. And so this higher number of this higher interface area matched with the better binding energy actually makes a lot of sense. And probably the more we cut, the more they will match to one another, because it ends up making sense. And probably if we did more replicates, it would happen in other of them. Okay, so as of right now, we have pretty much 4 concluding remarks. And the first one is that our protein is highly stable throughout our simulations. On top of that, both the knot and the catalytic triad are also highly stable, even when we start cutting our protein. We also end up concluding that was the contact with the ubiquitin that actually seems to induce the arrangement of our binding pocket that ends up leading to the activation. Once again, we can see this in the wild types, but also in the truncated variants. And finally, and as of right now, we can tell that our knot doesn't really seem to play an important role or in role in the protein's activation and the stabilization of the catalytic triad. In terms of future work, what we are doing right now, or we have already done, we decided to pretty much try to unknot our protein without cutting any of its residues. So we did this with a STIRMD simulation, and what we did is that we identified the residues that compose the loop that goes over our N-terminus. With those same residues, we end up defining their geometric center, that is here highlighted by this sphere. And then having this as a reference, we started pulling our N-terminus. We did this by first using the center, the geometric center, and the seventh residue. We pulled around 4 or 5 angstroms. Then we changed from the seventh residue to the sixth one, and then to the fifth one, until we were able to completely unknot our protein. And that's what you can see here on the animation on the right. So as of today, we have a complete protein with its 223 residues, completely unknotted. And what we want to do next is actually use umbrella sampling to study our protein and to be able to quantify the energy barrier that happens during the knotting process in the folding of our protein. Okay, so last but not least, I would like to thank both of my supervisors, all of my friends and colleagues, and of course, all of you that are here today hearing me. So thank you.
Thanks. Questions? actually, I want to. What is a Gordian knot?
It's a 5-2 knot. It's the name of the knot, just has a different name.
Okay.
Actually, it has a weird history regarding the Gordian name, but I really can't tell you the history. Sorry, I couldn't hear you. I can't hear you.
Which one of this?
Show the first slide.
We're going.
Just too long. I still have my question.
The Gordian knot is the 5-2. There.
Okay. Oh, you want to go first, Antonio?
Okay, I'll go first. I was just curious. At the beginning, you mentioned that in this case, the knot had probably the function, whatever, of making the access of that region more difficult. Is that believed to be a general role of knots or not? Or is it not really?
We don't really know what the role. What we think as of today is.
Okay.
Or not really do have a purpose, or they just don't really do anything bad to the protein, so just to keep existing.
Just happens.
Yeah, exactly.
Okay, thank you.
Okay, my question is something that we talked about before, so you'll have an opportunity to shine for the people that don't know. But it's regarding the folding of the knots. What we talked about before was, in your specific protein, what was the order that the loop was formed in the folding process. But now I was curious if what you explain and are going to explain for everyone else that doesn't know is the same thing for the other types of knots, or if each knot has a different folding structure.
I don't think that it's really regarding each knot, regarding each protein, because we have a lot of knots and a lot of proteins. And of course, I can tell you about this specific case. What we know today is that the protein actually folds and then the knot is formed. However, I can tell you that this happens all the time, but it is actually probable that this is the case, because it really doesn't make sense that the knot is formed first and then the protein folds to its secondary structure. So that's what we know. We don't really know a lot about knots, so maybe it is like this, maybe it isn't, but as of today, this is what we assume.
If you had to guess, because the 5-2 knot has a very specific structure.
Yeah.
If you had to guess for like the 5-1 or like the 6-1, would you assume that it would be the same, or do you think it would be different? Again, you told me it's protein to protein, but
yeah, but I think it would probably be the same, because it really doesn't make sense that the knot is the first thing to happen. It makes sense that it's the last one.
Okay, I don't know anything about knotted proteins, so.
Nor do I, but I'm going to ask whether
at the interface of the knots, whether you see certain kinds of residues, perhaps like glycines, which have more flexibility and don't stick out as much, or sometimes there's certain kinds of interfaces that have particular residue types in the interface, and at the knot interface, say you have small amino acids, or something that would make it, or maybe aromatic that are slippery, or
I really don't think that this is the case, and that is something that we can see. Let me just show you the slide. Because if it was the case, when we started deleting our residues, the knot would probably start to slip. That would make sense. But in this case, we can tell that even when we start cutting, the knot pretty much doesn't move. The end terminus doesn't slip at all under the loop.
Just to build on what Sara what she said is perfectly correct. I think there's a word that's the only thing that you didn't mention, but it's very important for the discussion, is the fact that it's a shallow knot. And what defines that, the fact that it comes before the collapse of the protein in the folding, or it happens after, it's because it's the shallow. Usually it's people think it's an entropic thing that happens during the folding, but when it's a shallow knot, there are some evidences that the knot forms after, and they're very radical on that. That's why we are studying it. And what these results actually show is exactly what Sara did. It's you can do very nasty things with the protein. You start cutting amino acids, and the tail stays there for, okay, it's 500 nanoseconds, multiple replicates, but still we don't see it move. So it's just like if there's like a highway over the end terminus, and the end terminus just doesn't care. So if we had to extrapolate from one case to the world of knots, we would say that, yeah, just there. Which maybe if we were looking for something very important, we still have to look maybe elsewhere.
Okay, then thanks, Sara, for the presentation.